There is no sudden edge where the familiar ocean ends. The change is gradual. Sunlight disappears, temperature falls, pressure rises. The water column keeps going down through distances that are difficult to picture from the surface. At the bottom of the Mariana system, the seafloor is almost 11 kilometers below sea level. Reaching it means crossing an entire vertical world. This depth did not open like a crack. Along the Mariana system, the Pacific plate bends and descends beneath the Philippine Sea plate. The process is subduction, one plate moving into the mantle beneath another. Over immense spans of time, that geometry produced a narrow arc of exceptionally deep seafloor. The trench is not a single point, and its deepest places are not all interchangeable. Maps can make the trench look like a dark line. A GEBCO bathymetric grid can help us draw its scale, but even a detailed grid is a model assembled from measurements and interpolation. It is not a photograph of every meter. The most honest picture begins with the limit of the picture itself, a vast landscape, sampled in fragments under nearly 11 kilometers of water. On the 23rd of January, 1960, the Bathyscaphe Trieste carried Jacques Piccard and Don Walsh into Challenger Deep. They descended inside a pressure sphere beneath a huge gasoline float. The journey took hours. Outside, pressure climbed to more than 1,000 times the pressure at sea level. Inside, two people watched a small circle of the deepest known seafloor through a tiny window. Trieste proved that human beings could arrive. It did not make the bottom familiar. The observers reported a flatfish-like animal, but that identification is widely treated with caution today. What the dive established beyond dispute was access. A crewed vehicle had touched the deepest part of the ocean. What it could observe was still limited by darkness, disturbed sediment, a narrow field of view, and a few hours in one place. Later vehicles replaced that single window with cameras, sampling arms, sensors, and repeated dives. Even then, every image remained a small frame cut from an enormous environment. In 1998, KAIKO Dive 0073 worked at a site JMSDC calls Mariana Trench A1, with a representative depth of 10,898 meters. Its archive includes an item classified simply as arthropoda. The location label matters. Mariana Trench A1 is the institution's wording for this dive. Calling every extremely deep Mariana record Challenger Deep would erase the distinction between a trench, a region, a named depression, and a particular sampling station. That classification matters because it makes a careful claim possible. An arthropod was recorded at extreme Hadal depth. The page separately lists samples of Hyendelia gigas, but that does not prove the animal in the video is that species. At this depth, honest evidence can be narrow and still be powerful. A moving body, recorded where pressure is crushing and food is scarce, is enough to overturn the image of a sterile floor. The bottom was not empty, but life there does not look like a crowded reef. It is patchy, adapted, and strongly connected to what arrives from above. Amphipods and other scavengers can exploit rare falls of organic material. Microbes occupy water and sediment. The Hadal zone begins below 6,000 meters, yet its ecosystems remain linked to the sunlit surface by sinking particles, carcasses, currents, and chemistry. One boundary is especially visible. In 2022, cameras recorded a fish at 8,336 meters in the Izu-Ogasawara Trench. It is the deepest filmed fish record. That is comparison evidence, not Mariana footage. The record helps show where vertebrate life has been observed, but it is not a universal line beyond which every fish is proven impossible. Records are shaped by observation effort as well as biology. Cameras cover tiny areas for limited periods, and an absence in the image is not automatically an absence from the environment. The safest boundary is therefore empirical. This is where fish have been filmed so far. Laboratory and field studies point to biochemical pressure limits involving proteins and compounds such as TMAO. Those results support a physiological explanation for why fish records end in roughly this depth range. They do not turn one measurement into a law of nature. More important, the end of the fish record is not the end of life. Below it, invertebrates and microbial communities continue into deeper water and sediment. To understand why, look upward. The trench receives material made far above it, fragments of plankton, fecal pellets, wood, and occasional animal falls. Slopes and earthquakes can move sediment laterally. The V-shaped topography and depositional processes can focus organic matter into Hadal sediments. This is not a perfect one-way funnel, but it is a mechanism that can concentrate traces of the surface at extraordinary depth. Every particle carries information. Carbon can become food, mineral grains record transport, molecules can preserve signs of biological activity. A trench is therefore more than a remote hole in the seafloor. It is part of a connected Earth system. The same connection that supplies energy also creates a darker possibility. Material produced by human society can follow pathways that were never designed to keep it near the surface. A global database analysis reported a plastic bag at 10,898 meters in the Mariana Trench. JAMSDC's KAIKO archive also contains a separate item classified as marine litter at Mariana Trench A1. The exact object in that clip should not be renamed without inspecting it. The stronger conclusion needs no exaggeration. Human-made debris has reached the deepest ocean. Visible litter is only one line of evidence. Studies of Hadal fauna from the Mariana-region have measured persistent organic pollutants, including PCBs and PBDEs. A different study traced methyl mercury in deep fauna to production in the upper ocean. These findings came from different collections. They should not be merged into one dive, one animal, or one object. Together, they show multiple routes by which surface activity can reach Hadal ecosystems. The bottom was not pristine. That does not mean every sample is dominated by pollution or that every unusual molecule came from industry. It means remoteness is not isolation. The trench can receive natural organic matter, terrestrial material, products of geological seepage, and anthropogenic compounds. The scientific task is to separate those sources, measure their effects, and resist the temptation to turn a complex chemical record into a simple horror story. Then the direction of exploration changes. The camera reaches the seafloor, but the investigation keeps descending. In Challenger Deep, researchers recovered a sediment core roughly 7.5 meters long from about 10.8 kilometers water depth. The core is not footage of an underground world. It is a vertical archive, layer after layer of sediment, chemistry, cells, and genetic materials sampled beneath the bottom. The 2026 study found microbial communities structured with depth. It recovered metagenomic evidence for pathways including hydrocarbon degradation, and cultured isolates demonstrated related metabolism. That does not mean the hydrocarbons prove an oil spill. The paper considers biological, terrestrial, natural seep, and human-associated sources. Genes show capacity. Cultures show that selected organisms can perform relevant processes. Neither result should be stretched into a complete picture of the buried ecosystem. Even so, the spatial idea is remarkable. At the deepest seafloor, life is not confined to what a lander can illuminate. It continues into sediment beneath the vehicle. Oxygen conditions change. Available compounds change. Communities change with them. The bottom is not a final boundary. It is another surface with a living record extending down through material that accumulated over time. The scale of the unknown becomes clearer when many samples are compared. The Mariana Trench, Yap Trench, and Philippine Basin were all included in the Mariana Trench Environment and Ecology Research Project, known as MEER. It analyzed 1,648 sediment samples. The sequencing total reached 92 terabase pairs, a metagenomic survey far larger than any single dive or core. From those multi-region samples, researchers reconstructed 7,564 species-level genomic taxa. 89.4% were reported as previously unrecorded in available databases. These are not 7,564 Mariana animals. They are not automatically formally named species. They are genomic groupings drawn from sediment across 3 regions, revealing how much microbial diversity still sits outside the catalog. Numbers that large can sound like an ending, a census completed, a hidden world counted. They are closer to a new beginning. A metagenomic sequence can suggest identity and capability without showing an organism's full behavior, abundance, interactions, or ecological role. Sampling is uneven. Databases are incomplete. A taxon that is new to a database may be common in a place scientists have barely examined. The unknown is not a blank, it has structure. Some gaps come from places never sampled. Others come from organisms that resist cultivation, genes with no assigned function, or short sequences that cannot yet be connected confidently. Better tools move the boundary, but they also reveal how much remains beyond it. The deepest ocean is therefore known in layers. A human arrival proves access. A camera proves that a body moved through one illuminated frame. A recovered object proves material presence. Chemistry reveals compounds. A core preserves change beneath the seafloor. DNA expands the inventory. Each method answers a different question, and each leaves a different darkness around its answer. Now reverse the descent. Rise past the sediment core, past the floor at almost 11,000 meters, past the deepest fish record, which belongs to another trench, past 4,000 meters, where the abyss would already seem extreme. The Mariana system falls away beneath an ocean that is still connected from surface to bottom by matter, motion, and time. What humans found at the bottom was not one secret object or one final species. They found evidence of life under pressure, the reach of human debris and chemistry, and biological information continuing below the seafloor. They also found how narrow every view remains. We reached the bottom. That did not mean we understood it.